I'm the project science coordinator of the EU-AIMS project, and I'm also going to lead on our new Phelan-McDermid syndrome clinical study. I'm very sorry I can't be with you in Orlando, and I would very much like to thank Geraldine and the Phelan-McDermid Syndrome Foundation for giving me the opportunity to tell you about our planned work in this way. So EU-AIMS is a large European public-private partnership that was launched about 2 years ago, and it includes 14 academic partners across Europe and 7 members of the European Federation of Pharmaceutical Industries and Associations, as well as patient organizations such as Autism Speaks and Autistica. Our consortium includes 5 integrated programs, including cellular assays and animal models, with a particular focus on synaptogenesis deficits that includes the Schenck family, but also neurexins and neuroligands, a translational neuroimaging program, clinical research development, and human genomics. Our overall goal is to identify biomarkers and pathophysiological mechanisms that aid in the diagnosis and prognosis of ASD, and also in the development of personalized treatment approaches. We have, for the past 2 years, conducting 2 large-scale clinical studies that span the entire developmental age span, from infants at risk for autism from 4 months through to 36 months, and another European-wide study that spans children and adults with autism spectrum disorder from 6 years through to 30 years. And our new PMS study largely is built on both our infrastructure, but also our experience with particular methods and tasks and techniques that we've developed for these 2 studies. So the overall aim of this new study is to create a comprehensive profile of each volunteer with PMS across different levels that includes clinical diagnosis, symptomatology and comorbidity, a neurocognitive profile, and here we mainly rely on eye tracking as a way to understand, for example, what a person finds interesting, and attention orientation, disengagement, but also brain structure and function using fMRI and EEG and MRI techniques, biochemistry and genomics. So our specific aims are firstly to understand the nature and the prevalence of autistic and other clinical symptoms in people with Phelan-McDermid syndrome. Secondly, we aim to identify biological risk markers at the level of cognition, brain structure, function, or biochemistry, and we are interested to see which of these markers might be specific to PMS and which of them could be shared with other forms of autism. Then we are interested to see why among the group of children and adults with PMS, some have ASD symptoms and others have not, or they differ in terms of their level of intellectual disabilities. And we are interested to see whether we can use biomarkers to identify treatment targets and also prognostic predictors for symptom severity or quality of life. Then finally, we will also carry out a complete genomic characterization of both your child and both parents where possible to understand the effect of multiple hits or genomic background on biomarkers and symptom severity. So I'm very fortunate to work with a great team here at King's College London. It includes Hannah Antonio, who has a PhD on low-functioning autism and learning disabilities, Daisy and Jess. So if you are interested in taking part in this study, one of our team will phone you for a screening interview, for example, to make sure that your child is safe to have an MRI scan, and also to get an estimate of his or her ability level. Because we realize that each child is different, and so the protocol will have to be adjusted to your child's age and also ability level. If you're still happy to take part in our study, we'll ask you to return a consent form and then send your login details to our online questionnaire, which you can complete at your own time at home. Then we'll ask you and your child to come visit us on 2 occasions. If you travel from far, our team will arrange for an overnight stay in a nearby hotel, and if you're living close by, you might want to come on 2 separate occasions. Each visit includes a number of tests for your child, but also parent interviews. So this is just an example of our online questionnaire platform, which probes, for example, for medical and psychiatric history, but also ASD symptoms in the different domains, but also other psychiatric symptoms, prenatal history, and other information. So then we have a small EEG and eye checking lab here, and we were very happy that our first pilot with a lovely girl with Phelan-McDermid syndrome was able to complete all the tasks. So I think it's a bit of a beginner's luck, but we feel emboldened that some of the children will be able to do it. So for our eye-tracking battery, we have strongly built this on our experience with the babies and younger children with ASD. So we have short sequences of 1 or 2 minutes at the most that do not require prolonged attention and no verbal response. They tap different cognitive risk markers of autism. So for example, a preference for or diminished attention to the eyes that's often been reported, or a diminished preference for biological motion over other forms of motion, or differences in attention orientation or disengagement. An example for our EEG task is the so-called mismatch negativity, which records the brain response to an unusual or odd stimulus in a sequence of predictable stimuli. And during this, while showing your child a video of Pangu, if he or she doesn't like Pangu, she's more than welcome to use her own video. And while she's doing that, we just in the background play these odd sounds. On an iPad or other toys, and again, we just let the sounds play in the background. We also carry out an assessment of autistic symptoms and cognitive ability, and those tests are geared to the intellectual level previously estimated for your child, and also carry out some interviews with yourself about, for example, level of social adaptive function. So then you go home, or you stay in a hotel overnight, and on the second day we'll ask you to come back to have an MRI scan, and we will do that under sedation or natural sleep. And also take some biological samples, so blood ideally to carry out genomic characterization, hair biopsies in order to generate induced pluripotent stem cells, and urine samples for biochemistry. Our extended Shanghai 3 team includes Professor Jack Price, who's leading on the stem cell work here at KCL, Professor Thomas Bergeron, who's leading on the genomics work package, and Tobias Sprackers and colleagues who are leading on the preclinical work. So together, this allows us to have an integrated program on Shanghai 3 that spans cellular assays through to clinical characterization. We will fund you for your travel expenses, and that includes the cost of a hotel if that's needed, and also we'll give you a gift voucher or a voucher for an activity to thank you for your participation. We'll also have regular newsletters and hope to stay in touch with preliminary results. So we hope to see you in London, and if you have any queries or comments or feedback, then please just email me. This is my email address, or the team. Thank you very much for your attention.
So this study is being conducted at King's College in the UK, and they are recruiting families in the UK right now. So if you're from the UK, you're welcome to contact Dr. Val and enroll in the study. Now we're going to turn our attention to what's happening in the United States, and I'm very happy to reintroduce you all to Dr. Joseph Buxbaum from the Seaver Autism Center at Mount Sinai. He is going to tell us about something very exciting, and the idea of bringing together our research community in a single consortium, and this could be a very fruitful approach to really being able to find enough patients to really make a difference in Phelan-McDermid syndrome research. So.
Okay, I'll make sure to talk about
And can I just say one more thing? He's actually worked tirelessly to make this happen. He's going to tell you about a grant, and it actually compelled him to go back and do a bunch of scientific experiments to make sure that this was really going to be a fruitful consortium that he and another researcher have proposed.
Good morning. It's good to see many of you again, and all of you. So, I'm the director of the Seaver Autism Center in New York City, and Alex Glebzon, who's sitting here, who's also one of our key people, I think, who directs all the medical clinical care, and medical research, has about half an hour, 45 minutes next to present. So we'll go into quite a bit of detail about prospective studies we're doing in the clinical trials, the attempts to develop kind of standard medical recommendations, and also talk a little bit about the creation of a network, which...
Joe, can you talk a little closer to the mic?
Sure. Some of the people at the dais are part of this new network. Eva came and met with us several times so she could be kind of brought up to date before they saw their first patient. So, the Seaver Center is built on the concept that academic medical centers need to be developing new knowledge, not just providing treatment. You have to figure out the next, what's the next best treatment that's going to come down the pike. And so the Seaver Autism Center is a research-oriented clinical center. It does a lot of things in various aspects of developmental delay syndromes, including autism and Phelan-McDermid syndrome, genetic testing, counseling, detailed assessment, developing plans for the children, medical management, social skills training, genetic research, and so on and so forth. And the idea with the Seaver Autism Center is that everybody gets together all the time, no matter how much they don't like each other. So it's pretty infrequent that you can get a hardcore basic molecular biologist in together with a physician who did not do any research during med school, and have them actually kind of over time evolve a relationship such that they can actually work together. And so we've kind of done that very systematically, I hope, or I think, for quite a few years now. And the result is that we have a real translational impact, so that we can go from an animal finding to a human finding. We can hear what's going on with the patients, and we can go back and say, do we see that in the animals? I'll give you a couple of examples about that. So this is the Seaver Autism Center team, minus a few people who were not available that day. So it's pretty big. There are a lot of trainees in here. We've developed a pipeline of philanthropy to support people who want to go into autism research and Phelan-McDermid syndrome research. We have people at all levels of their training, in medical school, in graduate school, doing their residencies, doing their postdocs, junior faculty, senior faculty, all trying to make a commitment to A, working together, and B, to focus on kind of this translational mission. And just to give you a sense of some of the directions, in preclinical research we have animal models, which I'll talk a bit about. We do a lot in cell and stem cell work. We have patient-based research on diagnostics and biomarkers, and neuroimaging, clinical trials, both medical and behavioral, and of course genetics. And then there's the clinical care arm and the training arm, which I won't get into, but I think is one of our most important things. Who's going to be the next generation of people in the field? And community outreach, bringing what we think we know to the greater public, and also listening to our consumers, which are the families, and asking what are the issues for them. So within Phelan-McDermid syndrome, I took over the Seaver Autism Center in 2008, and I was already kind of developing an interest in SHANK3. We began to make a mouse model, and so we had an opportunity there through some donations to say, we're willing to bring in families. We brought in over 40 families and do a complete prospective, very laborious, time-intensive characterization of the families. I think you all know that Phelan-McDermid syndrome is going to turn out to be, the data is already there, it just hasn't been fully integrated by people, as common as Rett syndrome, more common than other things that we know a lot about, and we know very little about Phelan-McDermid syndrome as compared to other disorders that have been kind of in the public domain for many years. So one of my jobs is to get the message out to the funding agencies. This is not such a rare thing, and we need to know as much about Phelan-McDermid syndrome in terms of course and effective treatments and so on as we do with the other disorders that I mentioned. And from my perspective, even more importantly, or equally importantly, develop the tools, preclinical tools, to actually come up with novel therapeutics that actually make a difference. And we'll get, and Alex will talk at some length on a clinical trial phase 1 is just completed. So, I don't know where I stopped there, but so we have both the animal models in Phelan-McDermid syndrome. We have 3 different mouse models. We have a new rat model, the first of its kind. We have stem cells from over 10 families, and we're here now with 2 coordinators to collect stem cells from additional families. Clinical research, I continue to look for additional mutations because they inform us about biology. We're doing a lot of work on biomarkers, some very interesting work about things that you can do to detect changes in the brain very easily in Phelan-McDermid syndrome, genotype-phenotype correlations, prospective phenotyping, and then also longitudinal, looking over time. And then, of course, the clinical trials. And we do think we'd like to become an expert site, and what Geraldine was mentioning, we've always felt, and it's just the nature of the field, it's much better to have an expert network, right? You want to be able to learn from others and share your, we're good at one thing, but Audrey's good at 10 things, and we want to learn from her, and vice versa. So we reached out to several sites. One of them was Lata Surya. Lata saw all the patients when she was working in the City Autism Center, and then she moved to Rush, so she's creating her own center, expert center there. We're working with Audrey. A couple of, we've obviously talked a lot to Eva in EU Ames, with Katie and many other people to try and develop a network of expert sites that all can bring their own experience with families and patients together, and to make clinical recommendations about what is a proper approach to treating individuals with Phelan-McDermid syndrome, and what are the things that are going to be most informative to develop better biomarkers and better treatments. So there is something called the RDCRC, the Rare Disease Clinical Research Centers. There were 19 of them. They come up for renewal every 5 years. And because Phelan-McDermid syndrome is a rare disease, it meets the criteria for being part of the RDRCs. Every RDRC has to have 3 or more disorders. So we have P10 mutations, Phelan-McDermid syndrome, and tuberous sclerosis, so it's 3 neurodevelopmental disorders that share some traits in common, and may even share some pathways, some brain pathways in common. And we apply to the NIH. These are very competitive. The people who have them do not give them up easily, not only because they want the money and they want their own interests to move forward, but also because there's a lot of politics, right? If there's an RDCRC in a certain state, that congressperson is going to try and protect it. There's a lot of behind-the-scenes things going on. Interestingly, the main funder of the NICHD was not part, and the NIMH was not part, the Mental Health Institute within the NIH was not part of the past rare disease networks. They're interested and want to be involved, and so a multi-site, that we put an RDCRC that has multi-site Phelan-McDermid syndrome and the 2 other disorders I mentioned, and it's still under consideration. We don't really know what that means. It's well past the deadline to decide. It means that there's some backroom politics going on, and we don't know how it'll fall, but we are committed. I mean, we were committed before we put the grant in. We already reached out to these other sites, and we will continue to do it, and sooner or later we'll get the funding to support it. But the process of developing a multi-site network is already ongoing, and we exchange protocols pretty regularly, and as well as patient samples. So just in the next 10 seconds, looking at, we have a study going on in 3,500 kids with autism. The biggest finding there is by far and away SHANK3. There are 7 kids with Phelan-McDermid syndrome, and we didn't completely cover all of the gene even. We have this new rat model, and I'll just make 2 points. One is they have very severe attentional deficits, which you probably recognize as something that's a core deficit in Phelan-McDermid syndrome. They have deficits not in social activities, but they have deficits in long-term social memories. They can make memories, they can retain them for a short time, but they have deficits in maintaining for long times. I'm not saying this is what we see necessarily in kids or in adults with Phelan-McDermid syndrome. I'm just saying it's a good biological tool to see what drugs are going to make a change in the brain. We see electrophysiological changes in the brain, and we've begun now to dissect the brains of these animals and do large-scale proteomics and RNA sequencing and look at every protein in the brain and see what changes. And we have a pathway involving actin dynamics that's profoundly dysregulated in these animals. And that just the way this thing works, and you'll hear about it more from Alex in a different example, there are drugs that regulate actin dynamics. So we can now manipulate the animal and increase and decrease actin dynamics in the brain and see whether that changes the behavior. Does it make the attention get better? Does it reverse the social memory deficits? So this is the preclinical space where we can really, having an animal or a cell model gives you the power to do a lot of things you obviously aren't going to do and anything else. Alex will talk about the fact that we've done something similar with IGF-1 that led to a clinical trial, and I think I'm going to just stop there and thank you all for your attention.
Keep questions for the end of the day. At 5 o'clock today, we'll have a Q&A session. I'm delighted to introduce you to Dr. Coloson, who is, you're not going to say anything more? Okay, you're just going to wait until, okay, that's great. So I think Dr. Thurm is next. Dr. Thurm is an expert from the National Institutes of Mental Health. She's a role expert in autism research, and she's one of the groups that's working in collaboration with the other sites. And maybe you know.
Good morning. Thank you all for being here. We're very happy to be here. I think I'm going to start out by talking about NIMH. Now that I know this will be videotaped, I'm going to be very, very cautious about what I say. But to just explain, because it's hard for me to even understand, that NIH has two different total entities. One of them is extramural, and that's where most of the money goes, and that's to funding grants. And the other is a small portion of the money goes to research in the intramural program, which is in Bethesda, Maryland. It's I'm going to show you that actually. And so we are here as intramural researchers, people who are doing science, again, that gets funded, but separate from the funding of all the other places in the world. So within intramural, I am part of a branch that's called Pediatrics and Developmental Neuroscience Branch. But I'll already say that even though pediatrics is in the title, we do hope and plan as the whole network to be studying individuals with Phelan-McDermid across the lifespan. So don't get bogged down by pediatrics. Because it's development that we want to look at across time. And the mission of our branch is to identify factors that influence the onset and expression of behavioral symptoms, to develop effective treatments and prevention strategies for disorders that start in childhood. This includes autism and neurodevelopmental disorders that relate to it. So this is who we are, and I think just like all the teams that are involved in the natural history studies, it is truly a multidisciplinary effort. And it's not just meeting with lots of these different individuals separately. It's a cohesive team that hopefully will work literally in the same room with you so that we're all saying the same thing, even though we'll be looking at different aspects of development. So I think just by looking at our team and who the individuals are and what disciplines they come from, that helps you, again, think about all the different aspects of the child, the individual that will be studied, looked at, examined, and hopefully treatment recommendations can be made both for the short term, and then as we learn more things about the phenotype and characteristics, we can learn to plan more for treatments in the long term. So I'm just going to whiz through and just show you that this is what is called, because everything at NIH is research. There is no just clinical care that is done without research. It's not called a hospital, it's called a clinical center. And this is the picture picture of it in Bethesda, Maryland, and I also wanted to just show you that NIH is lucky to have what we call the Children's Inn, which is a separate facility where individuals coming from far away can stay, and it does have a really cool playroom, actually multiple playrooms. So there are places where patients can go and have downtime when they're there if they're coming from out of town, especially because it will be several days of different kinds of testing. So NIH and our group didn't think about Phelan-McDermid, out of nowhere. NIH does have a long history of conducting natural history studies on specifically rare disorders, and more of these disorders have been rare neurogenic disorders that affect neurologic symptoms. And so we think it's really important to study Phelan-McDermid in the context of some of the other disorders we have looked at. And so just to examples, I'm sure folks have heard of some of these that have similar in terms of early onset, overlap with autism symptoms, intellectual disability, motor impairments, and then other medical issues. And that's exactly what we plan to do, just like the other studies you've heard about. We plan to have individuals have clinical neurologic examinations, and that includes neuropsychological, IQ, language, a lot of testing in terms of communication and language, as well as other symptoms relating to autism and neurodevelopmental behavioral problems, MRI and EEG, and then consultations with appropriate services. So these would include things like rehabilitative medicine, which includes physiatry, occupational therapy, physical therapy, speech therapy, nutrition, neurology in terms of looking at sleep as well, gastrointestinal and renal. These are just some of the areas. But I think the idea is that as we're learning more about the phenotype and characteristics, we will add on and individualize as we need to. And that is the model. The model is that you conduct these evaluations. In our case, we don't want to do genetics in isolation. We want to do it in combination with the other groups. We want to do things like conduct MRIs that are not just individual results, but also can be used across the sites to look at. And there's a lot of methodology involved in making sure that we can share this data in a meaningful way. So we want to, again, combine the data across multiple sites. But on the other hand, we also want to be able to compare the data across multiple disorders, because I think that's how we're really going to learn what's most unique to Phelan-McDermid and what's not unique, and that will help us with the biological pathways that we're looking for. So I think with that, I will end. I think you're hearing from the same thing in different ways from people, which I think is important to know about how these natural history studies will be conducted and how important the data is and how it will be used. Later today, I'm giving another talk, and I will provide contact information there. Okay? All right.
You next on the agenda. Okay, so we'll let Lindsay go. Dr. Oberman go next from Harvard. And many of you may know her because she's been doing phone interviews, so you may have done like a 2 or 3 hour long phone interview with Lindsay a few months ago. Thank you. I am thrilled to be here, talk to you all a little bit about what we are doing at Children's Hospital in Boston. I did have the opportunity and privilege to talk to some of you all over this past spring. I'm sorry for those who I wasn't able to get in touch with. It's a really good problem to have when we put a recruitment ad out, and I got literally close to 200 families respond, and there's only 24 hours in the day. So I really, really appreciate, yes, applause to you all. Thank you very, very much for your dedication to research. I really, really appreciate you all. So I was able to speak with a number of you all, schedule interviews with 40 families, and I'm going to share some of the preliminary data that we have analyzed so far, and to tell you a little bit of where we're going from here. So to give you a sense, we are based in Boston, Children's Hospital, and I work with Dr. Walter Kaufman, who is a pediatric neurologist there and who heads up a clinic where he sees patients with Phelan-McDermid syndrome, along with Rett syndrome and other autism patients. I'm, in fact, an experimental psychologist, so I work with Walter to head up some of the research related to psychology within his clinic. So as I mentioned, we were able to do interviews with 40 parents, and so far, I'm just going to share a couple of the interesting findings that have come out. So we did an autism diagnostic interview, which is one of the gold standard diagnostic measures for autism. And what came out was about 65% of the children met criteria in the 3 domains. This was actually based on the DSM-4 criteria for autism. So there were 3 domains: in social, communication, and restricted and repetitive behaviors. So about 65% of the children met criteria in all 3 of the domains. So would indicate, and again, when you're getting a clinical diagnosis, you typically would do more than just this one interview, but is indicative of a diagnosis of autism. Then about another 15-20% met criteria in the social and communication domains, but did not show significant repetitive or restrictive behaviors. And then another few met in communication and restrictive and repetitive behaviors, but not social. And only 2 or 3 children out of the 40 did not meet any criteria in any behavior that was characteristic of autism. This is, I would say, consistent with the literature, that a majority, or maybe half to 2/3 of the participants would meet criteria for autism, but not necessarily all. And so my interest was to go a little bit deeper and to see what types of behaviors were these children showing, and how were they classified. So a couple things that stood out in, again, these just preliminary analyses were the expression of facial expressions. So a majority, about 75% of the children showed a limited range of facial expressions, displayed a limited range of facial expressions. The good news is, oftentimes, if there was only one facial expression, it was a smile. It was happy. The children were almost very generally described as happy children. And so that is great to hear. So if you're going to have a limited range, I'm glad to hear that it's happy and smiling. But that's interesting in terms of how social communication and exchange of emotions are displayed in these children. Another interesting, and I'm sure very familiar with you finding, is that the majority of the children showed obsessive behaviors in the form of chewing. And the question I had that we're going to look a little bit deeper in is, why were these children needing to chew? Is it a sensory-seeking behavior? Was it a stereotyped motor behavior? How should these types of behaviors be classified as they pertain to autism-like characteristics? Interestingly, few children showed certain behaviors that I would say are characteristic of children with autism. So this would sort of suggest that, at least in these children, they don't show these, in general, don't show these types of behaviors. And I always say in general because a few participants did, and every child is different. But specific ritualistic compulsive behaviors were rare. A circumscribed interest in a specific domain was rare, and an unusual attachment to objects was relatively rare. The other thing that stood out to me was in the question of a regression. A lot of the families said, well, they had certain skills, but then they lost them. And when I probed a little bit deeper, it seemed like
they lost them at times when they were transitioning maybe to other skills. That maybe it was a problem in remembering those skills or holding on to those skills or being able to pull those skills back after you've sort of disengaged the attention on them. And so there's a question of whether these children do show what we would call a true regression versus a variable skills in certain domains. So some future directions. In our next stage of this study, we're going to be bringing in families to come to Boston. We're working on specific funding for that to pay so that you all wouldn't have to pay for your travel and lodging. Also, with all studies, we have to go through ethics board and IRB process to make sure that the study that we're proposing is ethical. And so we're working on that paperwork and hopefully to get it approved in the next month or so. So I will be contacting you all or putting out another request if you are interested in participating in this next stage. We'll also be looking at the relationship between size of deletion, where the deletion is, genetic measures, and also epigenetic factors, things like expression of genes, methylation of genes, things like that I'm sure you'll hear more about, and the relationship between those and behavioral symptoms. And also as part of a bigger project, we are including neuroimaging and EEG measures. I'd like to thank you all for your participation again, and also the Simons Foundation for our funding. Thank you very much.
Proposed RDCRM network, and if it's not funded though, you still independently plan to continue with these PMS studies, correct?
Absolutely. No, that should be asked, absolutely. The question is just how much we would be able to compensate you all for your time and the extent, the size of the study that we would be able to do given the funding. But yes, we are dedicated and very passionate about working with you all.
Thank you. Okay, so Dr. Mosconi is from UT Southwestern. They recently began bringing patients from the Dallas-Fort Worth area into the clinical research study, and he's seen several. I'm not sure how many now. I'll let you talk about that.
I foolishly included a video in my presentation, so there's a 50% chance that it'll work. It's not much, but it's fun. Okay. This takes a lot of coordination. So first of all, thank Geraldine and the foundation for inviting me here to present information or present sort of a status update on what we're doing at UT Southwestern in terms of our Phelan-McDermid studies. This is really sort of a wonderful opportunity. I have to say that we'll have a table later where we can talk about more specific details of our ongoing research projects, but really the primary reason for coming down here is not only to pique your interest in our research studies, but to actually get to meet and interact with you all. So please do come up and introduce yourself as you've seen around the meeting, as well as Craig Powell, who I'm going to embarrass in just a few minutes. So our studies at UT Southwestern are relatively new in terms of the clinical studies, but these are taking place within the context of our new Center for Autism and Developmental Disabilities. It seems like everyone up here has sort of developed these studies within the context of a center. This is really sort of a critical component of what we do, both in relation to the Phelan-McDermid work, but as well as the work we do with autism and intellectual and developmental disabilities more broadly. This is an endeavor that we've undertaken through the resources at both UT Southwestern as well as in collaboration with Children's Medical Center and UT Dallas Callier Center. The reason that's important is it really brings to the table experts across various disciplines. You can't do this work sort of in isolation. You can't do this work just relying on your own smarts, because none of us are smart enough to really sort of take care of each aspect of what's needed to make progress in this area. So this center was developed to integrate the patient care as well as the ongoing cutting-edge research that we believe we're doing, as well as Dr. Buxbaum talked about earlier, the really big critical components of training people that will go out in the community and be more aware of what PMS is, pediatricians, psychiatrists, neurologists, as well as developing these community partnerships, such as with the foundation, with other groups that are going to help guide us in terms of directions and tell us what's needed in terms of for families, which is just sort of a critical component of what we do. The research that I'm going to talk about is really, there's an emphasis on the translational aspects of it. And you've probably heard this term now multiple times, even just within this morning. What I'm talking about when I'm describing or referring to translational research is the emphasis on integrating what is happening in a basic science lab where they're studying cells and animal models of PMS. And integrating that with our ongoing clinical research studies. We have to translate back and forth if we're going to really make significant progress and rapid progress in terms of identifying what are the targets that we want to aim at when we're developing new treatments, and how are we going to move the work that we do in the basic science labs to actually impact how we're both assessing and then treating individuals with PMS. This takes a rather large group of people to do it. So I guess we all had pictures of our team, so to an extent maybe we're all kind of social people, at least outwardly. But this really does involve a team of people that are both highly motivated and dedicated to this work, but also highly collaborative. So Craig Powell, you can see there, he's really been sort of the primary engine, I think, in getting these studies started. And Craig is a neurologist and basic scientist who's been studying Shank3 mouse models for several years now, and has really just sort of been tireless in getting these studies up and running, both at the preclinical end, the animal models, as well as at the clinical side of things. So I'm very, very grateful and lucky to be around Craig on almost a daily basis. He might not be saying the same thing about me, but he's stuck with me at this point. And then the rest of the team, I'm not going to go through them individually, but certainly if you guys are part of, want to become part of either the studies at UT Southwestern or come through for clinical care, I showed these other individuals. It's just a wonderfully dedicated team that I'm proud to be a part of, that are really providing sort of comprehensive care and are highly focused not only on autism in general, but also on PMS. So the aim of our clinical studies is really to develop a more comprehensive understanding of the behavioral, the cognitive, the medical issues that are associated with PMS, and understanding the course of these different challenges as they change over the course of an individual's development. We really are sort of aiming to do this work because we think it's going to guide us in terms of identifying new targets for developing treatments and for actually for testing those treatments first at the preclinical stage of things, and then moving those forward into clinical trials with patients. Participation in our studies is right now it's a 3-day affair that includes the medical and genetic testing, some of which has been is very similar to what you've seen in the previous presentations. We do an extensive battery behavioral and cognitive tests with each individual to really sort of map out where the strengths and the weaknesses are for each individual, and then we're also doing I think sort of more individualized and unique tests using electrophysiology EEG measures as well as movement testing, because we know that the motor difficulties in individuals with PMS can be quite profound. I'll show you a little bit of those to give you sort of a visualization of what we're doing. So the EEG studies, EEG is really sort of a unique form of measuring brain function and brain physiology in that it's non-invasive, there's no injections or anything like that. Individuals can stay awake, we don't need sedation for it, and it gives us really precise measurements of how the brain is functioning either at rest or when we present different stimuli to people, and really is precise on a temporal scale, so the timing of the different changes in brain function are very precise in EEG relative to MRI, and we now have new procedures for making it spatially precise as well, so we can map some of these waveforms onto specific regions of the brain and really get a better characterization, richer understanding of what actually is happening in the brain. This is data that I'm showing. I'm not going to go into explicit detail about the data, although I'm happy to talk more about it with people individually if they have questions. But this is just sort of an exciting finding that we've identified on just a few individuals with PMS at this point, and it's in response to auditory stimuli that are presented as individuals passively hear these tones. And what it's really showing is there's actually quite a profound and different neural response to auditory information or auditory stimuli in individuals with PMS. We'll see if this is true for all individuals with PMS or just a subset of individuals. But what you see there at that P2 point is that there's a profound reduction in that positive going spike or response 200 milliseconds after the auditory tone, which is involved in really sort of the processing of that sensory information. That seems to be distinct, and what's exciting is we're also moving these studies into the animal model studies, so we can look at how these electrophysiological responses may be impacted in the animal model and may respond to different treatments. The mice wear much smaller hats. So we also are doing motor studies, so these are sort of precise or quantitative analyses of the movement challenges or issues that individuals with PMS show. These look similar to, they're not quite as cool, but often what you may have seen when you've seen videos of people developing like different games and things like that. We're not as sophisticated as gamers, but we try. And so what happens is there are these markers that are placed on individuals, and then we can measure where those markers are in space, how fast they're moving, and how well they're really coordinated as individuals walk, as individuals reach for different objects, and then we also have force platforms embedded in the ground so we can look at sort of how individuals are balancing and moving around as they're just standing still or sitting for that matter. I do need to put the on. This is sort of the climax of the presentation, right? To see if this works. It doesn't, but I can get it to work. Give me a second. That's the one. All right. So this is just a presentation of the kind of information that we're actually getting from these markers and from this motion analysis. As you can see here, these markers are used to track the individual's movements over time as they're walking. The 1 and 2 on the floor, those are the actual force platforms, so we can get information in terms of where they're pressing on their feet, how their balance is maintained while they're walking or while they're standing still. So we can use this information to look at the velocity, the duration of different movements, the coordination. And what we've seen is very different movement patterns in individuals with PMS relative to age-matched individuals who do not have PMS during walking and during just sitting or standing. And this gives us insight not only into what may be useful therapeutically for these individuals, but also what may be happening at the neurological level, because we know quite a bit about how movement is controlled in the brain. And so this gives us insights into where in the brain, which specific circuits may be affected in autism, or in PMS. So for our studies, we're looking for individuals between 3 and 35 years of age with PMS. We're looking for individuals that are medication stable. I can talk to you more about what that actually means as we talk individually. We basically just don't want people moving between different medications during the course of the studies, because that can make interpreting the data difficult. We do cover local travel at this point. We're looking to cover broader travel, and local is loosely defined. All participants will be compensated for the time. And I think what has been helpful for the participants that have come through so far is we do provide a clinical report of all of the findings that we have. We wouldn't provide a report, say, of the movement findings, because they're very specific and non-standardized. But all the standardized tests that we do put in report format and then meet with families at the conclusion of the study. And this is how you can get in touch with us. Again, come up to me personally. I implore you, and we can talk more about details. But this is the contact information for us for those of you that are interested in participating or just want to talk about what's ongoing, either from a clinical or from a research perspective. So thank you for your time.
Isn't it great that we have all these various clinical research studies going on now in Phelan-McDermid syndrome? There are some other studies going on. In fact, we're going to hear more about a study that's going on at Stanford. We'll hear about that right after lunch today. And at the end of the day, there's going to be about a talk about epilepsy, and there's also a study associated with that that's happening through Baylor. So be sure to come back for those sessions to learn more about what's happening with clinical research studies.